AQUABIZ, all the best knowledge for aquatic system.
Yes, good morning, everyone. It is wonderful to be back here in Thailand. I came here as a young man 35 years ago, early in my aquaculture career. I came as a tourist, but as part of the tourism, I was also looking at aquaculture. I actually came here for the second International Symposium on Tilapia and Aquaculture, which was held at the Ambassador Hotel, not too far from here. So it's really wonderful to come back. It's been many years since I've been to Thailand, and I love your country very much. It's beautiful, as well as the people. I appreciated Lucas's comment about food safety, and in particular this issue of antibiotic residues, which is a serious marketing issue. It's a reputational risk for producers. And so really the underlying theme for this presentation is how can we produce shrimp without the use of antibiotics? So that's really what we're trying to look at here. So as a starting point, if you look at this graphic on the left, I'm going to try to point to both sides here. There we go. So this graphic on the left shows some of the different pathways by which antibiotics can enter into the environment. And it can happen in human use and in agriculture, and in particular farm animals, especially the waste and particularly the sludge. And so, certainly shrimp farms are part of this use in animal production. And this graphic on the right here sort of illustrates the problem. This was a research study that was done in Vietnam, and the investigators went into the canals, the discharge canals, and they were able to isolate 41 resistance genes. These are genes for antibiotic resistance that they were able to isolate. Okay, so this kind of illustrates the magnitude of the problem. And in particular, some of these are antibiotics that are used in human health. So ultimately, this comes back to being a human health problem. I think there has also been, more recently, an appreciation of kind of this bigger role of, the gut microflora, the bacteria, the microbiome, microbial community inside the shrimp gut, and its role in regulating the immune system as modulated by external factors like water quality, the microorganisms that are present in the environment, the kinds of feeds that are used, and management, in particular how you can reduce stress. I think there's also been an appreciation for the different roles that these beneficial bacteria can play. I've listed a few of these here. They produce these micronutrients like vitamins and short-chain fatty acids. They can help regulate shrimp digestion through the production primarily of enzymes. They can inhibit pathogenic microorganisms, and they serve a role in modulating or regulating the immune system. So, having a little difficulty. Yeah. So with that as an introduction, this is an overview of my presentation. I want to spend a few minutes to talk about the microbial communities inside shrimp ponds, focusing particularly on the vibrios. I think vibrios are greatly misunderstood in shrimp ponds. And then I want to go into a list of tools that we have available to us as producers, as shrimp producers, that could be applied depending on your context to control vibriosis. And you know, when I was talking about controlling diseases, I'm not really going to talk about viral diseases because you know that's another level. You really need to have very good biosecurity and good genetics to control viral diseases. But really, I'm going to focus in on bacterial diseases, primarily vibriosis. And then the last part of the talk, I want to provide some kind of practical applications of this information to have bioremediation in the pond. Okay, so this picture here is a depiction of a microbial community in a shrimp pond sediment. Okay, now it's not important to know what the individual bacteria are, but just to get a sense that these communities are very complex, okay? And there are these underlying questions here. So how can these communities be manipulated to the benefit of the farmer? And how is it possible to manage this assembly? If you look at this, it presents a picture that would suggest to you that it's very difficult to do that. And just, you know, as a point of reference, you've got basically bacteria, and the green ones are eukaryotes, like algae. And you'll see different colored lines between the bacteria that represents interactions. The blue lines indicate positive interactions between species. The red lines indicate negative interactions between species. So you can see the entire picture is extremely complex. Okay, so really what you have then is a situation where you have species. Now in the terminology, these are called OTUs. Basically, they're operational units. You can consider them species. And there's certain bacteria that are shared between water and sediment and the intestines of the shrimp. So if we consider that and we take that complexity that was in the previous figure and we try to reduce it, we can use a figure like this to reduce it to 2 dimensions, that previous figure basically. And what I want to point out here is that the microbiome of the sediment is very similar to that of a healthy shrimp, okay? The distances between these two are not that great. Well, this is probably not a big surprise. You know, we know that shrimp, you know, particularly tiger shrimp, but this is for white shrimp, I believe, live in close association with the sediment. You can also see that there's a difference in the microbial community between a healthy shrimp and diseased shrimp. And of course, there's actually quite a big difference between healthy shrimp and pond water, so it doesn't seem to have as much of an influence on the microbial community as does the sediment. So there are a number of factors that influence the microbial community in the sediment, and again, the sediment has been studied very well as the intestines. What this figure shows is that the microbial community shifts during the crop cycle. So the community that you have in the first month is not the same community you have in the second month, or the third month, or the fourth month, or the fifth month. It basically changes over time. Not in any kind of predictable way, but it's just changing. It's extremely dynamic with respect to time. It's also influenced strongly by conditions in the sediment, in particular, the amount of oxygen that you have. Of course, the oxygen penetrating into the sediment is going to affect the kinds of bacteria, broadly speaking, into aerobic bacteria and anaerobic bacteria. So that's a strong force, as well as the organic carbon concentration. So those are important drivers of the sediment microbial community as well. And this figure is actually very interesting because what this is saying is that basically 14% of the variation in the microbial community assembly can be explained by environmental factors, things like oxygen and temperature and salinity and things like that. A small amount, 10% or so, is explained by geographic distances. That's a farm location here, a farm location there. But here, look, 75% of the microbial community is unexplained. And what that suggests is that random processes are much more important than these deterministic kinds of processes like the environment and geographic distance. So the implication of this is that practical manipulation of the community is extremely difficult, okay, as a practical matter. So all this, you know, these last couple slides were really to show it's very hard to do this. And then we have a whole host of other factors that will affect, this is now looking specifically at the intestinal microbial community, and I just want to go through a few of these. So if we start with development stage, if you look at this figure here, you don't even really need to know what those species are. This is called a heat map, and the darker the color, that just means more of that particular bacteria. So you can see here larvae, juvenile, and adults, and you can see the community of larvae is distinctly different from that of juveniles, which is distinctly different from that of adults. So as the shrimp grow, the microbial community changes. And again, not in any kind of predictable way. Then you have a host of environmental factors that can affect the community composition, and these are really basic factors, things like temperature and salinity, toxicants like hydrogen sulfide that might be in the pond, starvation of the animals. And then this one is really interesting. This one is, I have this word up here. This is even an English word that a lot of people don't really understand what it means, but dysbiosis is a condition of imbalance, okay? So in healthy humans or in shrimp, the community is more or less in equilibrium, in kind of balance. And when the animal is sick and diseased, that community composition shifts. It tends to be less diverse, it tends to be dominated by fewer species, and that condition is called dysbiosis. And then there are a whole host of diet components. So, you know, really the feed formulations are going to have an important effect on the microbiota of the intestine as well, fatty acid and the different carbohydrate sources, including soybean meal, presumably. Okay, so now I want to talk a little bit about Vibrios. And again, I believe Vibrios are kind of misunderstood. So this is a picture or a representation of the microbial community composition in a shrimp pond sediment. This is not going, you're not going to see this all the time, but you're going to see something like this all the time. And if you look at it at the level of the phylum, which is a very high order classification, you're going to see that the sediment is dominated by proteobacteria. Now, proteobacteria are very common in wastewater treatment systems. They're common heterotrophic bacteria. And if you look at the genus level, you see that one group that is real prominent here are the Vibrionalis, and they are, of course, where the Vibrios reside. If we look at the species level, one of the more common species that you'll see in this particular sediment is Vibrio harvii. Well, most of you probably know that Vibrio harvii is a well-known pathogen, potential pathogen in shrimp farming, often considered just undesirable. Well, what this is saying is that Vibrios are very common in shrimp pond sediments, okay? So, you know, we have to kind of live with them. So Vibrios are an example of what are called opportunistic pathogens. And what are opportunistic pathogens? Are bacteria that don't normally cause disease. But when the animal is stressed in some way, if you have poor water quality, low oxygen, bad salinity, bad temperature, hydrogen sulfide, then they can cause disease. And also the bacteria can become pathogenic. They basically are not necessarily pathogenic sometimes, but other times that they are. And also, these opportunistic pathogens are not just Vibrios, but there's a whole list of other bacteria that can cause disease and that are potential opportunistic pathogens. Now what happens is that there's some kind of disturbance, and that can cause stress in the animal, and then that ultimately will lead to the disease basically by the activation of genes. Basically, genes turn on in response to this pulse disturbance and lead to the Vibrio becoming pathogenic and causing disease. And the way they do that is with a system of communication that's called quorum sensing, okay? And the way quorum sensing works is it's basically a way for bacteria to talk with each other. And they put out chemicals. They say, hey, I'm here, I'm here. And so you have these signal molecules here, and there's different kinds of signal molecules that are out there. And when the bacteria are present at low numbers, they can't hear each other because there's just not very many of them. And typically when the population density gets very high, they start hearing with each other, and these receptors basically cause the activation of genes that turn on, in this case, for virulence, for becoming pathogenic, for being able to cause disease. Normally that bacteria, like Vibrio harveyi, doesn't cause disease. But with this quorum sensing communication system, they hear each other, and when it gets to a certain density, they change their form, okay? And that's called I have it here. That's called phenotypic switching right here. So phenotype is just in this case, it's the pathogenic or non-pathogenic, and it can switch between those two modes depending on the conditions. Now, Vibrios are kind of interesting, or at least the one that causes EMS, AHPND, and that is that the free-living planktonic form is what causes disease. The biofilm form does not cause disease. It's not pathogenic. Usually it's the opposite with respect to many diseases. But in the case of Vibrio parahemolyticus, the non-virulent phenotype is the biofilm type. Now, the other thing about Vibrios, and this is really more conceptual. There's not been a lot of work done specifically with shrimp pond intestinal biota or sediment biota to show that Vibrios have this role, but by implication, I would suggest that they do. And that is one of a microbial gatekeeper, okay? A microbial gatekeeper is a bacteria that is basically keeping the system together. It's keeping it in balance. And if you remove that species from the system, it begins to become unstable. And there's a disequilibrium that occurs. So when you have these gatekeepers present, it keeps the whole community together. And these opportunistic pathogens are so-called keystone species. And keystone species are the ones that keep the community together. You take them out, and the community becomes fragmented and unstable, okay? And that leads to this condition of dysbiosis that I mentioned earlier. Okay, and finally here, just talking about Vibrios, we can almost look at this in a diagnostic way, although there are much easier ways to diagnose vibriosis. But if you look at the intestines of healthy shrimp, they tend to be a diverse community, okay? And in this case, they're dominated by these rotobacters, and I'm going to talk a little bit more about these a little bit later. So 60% of the community is these rotobacters. You know, there's some shared species, and in the diseased shrimp, the system is very simple, and 95% of the species in the gut of diseased shrimp, in this case, have Vibrios in them, okay? So healthy shrimp have rotobacters, diseased shrimp have Vibrios. And you see this even with other diseases, even like white spot. You know, you'll see a difference in the microbial community between healthy and diseased shrimp. All right. For this next part now, I want to talk about some of the tools that we have available that can help us, you know, based on this understanding of the microbial community, to support shrimp health. So let's take a look at those. And before I get into that, however, I would like to just emphasize the role of a health management plan. I really think every farm should have a health management plan written down, okay, with all your standard operating procedures related to shrimp health. Indeed, the BAP certification system, as does many others, requires this, okay? So this is a good idea with or without certification, but if you're going to do certification, you're going to need this. And in that, you want to look at your biosecurity and animal welfare practices, what you're doing to maintain good water quality, to reduce stress, what kind of feeds are you using, what your pond preparation protocols are, how you're evaluating the post-larvae that you're stocking so that they're clean. All of those should be spelled out. And you should have SOPs and record keeping to support this health management plan. So then I've listed a whole host of other tools that are available. We don't have time today to talk about all of these, and some of these are actually pretty interesting. We've got things coming down the road on quorum sensing disruption and phage therapy that are really interesting. And I even included chemotherapeutics in this list, but really I should just cross it out. It's a tool, but we really don't want to use it. We want to use alternatives to chemotherapeutics to support shrimp health. Okay, and one of the ways that I like, and that's kind of why I put this at the top list, is to use what I call microbially mature water. And basically that's water that you've used and that you're holding and reusing over and over again. And that's based on ecological theory. And that ecological theory applies to ecological communities of all kinds, including microbial communities, but terrestrial communities, other aquatic communities. And that is this idea that you have succession. If you start with just absolutely a clean pond, okay, and you bring water in that's been sterilized with chlorine, and you look at how things change over time, you see a succession pattern that occurs. And that succession pattern looks like this, where you have a bloom of these so-called R-selected species. And let me go back here. So you can really divide these bacteria into two broad groups of organisms. You have these so-called R-selected organisms, and R-selected organisms have a very high growth rate, and as a result, the populations grow quickly. Vibrios replicate every 15 or 20 minutes, so there's a new generation, very rapid. And these are the opportunistic pathogens like we've been talking about, like the Vibrios. In contrast, there are other groups of bacteria, these K strategists, that grow much slower, okay? But over time, they can outcompete the Vibrios and become dominant. The Vibrios don't go away, but they're not as important as they are early on in succession. So yeah, this is the pattern you see. You see this bloom of R-selected species like a Vibrio, and then over time, these slower growing K-selected species become dominant. K sort of refers to carrying capacity here. And the Vibrios are present but suppressed. Now, if you think about EMS, and the name early is in the system, of course, it's early on in succession. Here's what's happening. You fill a pond, you stock your shrimp here, and this is where EMS comes in, 30 days, okay? So right when you get that bloom of these opportunistic bacteria. So if you wait longer, this is a better time to stock the animals, okay? Because the Vibrios won't cause disease in that case. Now, you can accelerate that process by inoculation, using probiotics, using water from a previous culture cycle that has bacteria in them. And that's a way to kind of use this application of ecological theory to avoid using, avoid the risk of having a high density of potentially opportunistic pathogenic bacteria early on in succession. So again, you prepare the water ahead of time and before you stock the shrimp. Don't stock in clean water. You got to make that water a little bit dirty, and it's going to be safer water to stock, at least with respect to Vibrio. Okay, one of the other tools that's available to farmers is biofloc systems. I've done quite a bit of work with biofloc systems over the years. My thinking about them has changed a little bit, maybe has gotten a little more subtle. I think early on when biofloc systems were promoted, people were looking at trying to shift the entire dynamic of water quality in the pond over towards biofloc. and by a lot of addition of organic carbon like molasses. But really, I don't think that's a good idea. There's a lot of risks associated with that, and adding organic carbon like simple sugars is also, fuel for Vibrios as well. So, it's kind of counterproductive in many ways. But there are a lot of benefits to water quality and to what's going on inside the animal, particularly with digestion and immunity. And then you can set up some competition between beneficial bacteria that are present in the biofloc and these pathogenic microbes. So there's a lot of advantages to using a biofloc. Now, the biofloc largely, at a large scale, consists of all these things, but it also contains a lot of this whole list of things here. And a lot of those are very favorable for immune stimulation and to aid in digestion and so forth. So there's a lot of really positive aspects to having biofloc. Anyway, to return to my previous point, and I didn't kind of drive the point home, and that is that I think having a lighter biofloc, not so heavy, more of an autotrophic biofloc, I think that is much more beneficial than having this heavy floc where you have in the cone where you would have like 15 to 25 milliliters per liter. You're more looking at something between 5 and 10 or even less than 5 in some cases. Not a lot of floc, but having some floc there is beneficial. Okay, so I want to talk to you about some really interesting research that has gone on in the last couple years. And this was an investigation done in aquaria in a laboratory. And basically what they did here is they grew shrimp in autotrophic or heterotrophic bioflocs with or without probiotics. So here you have autotrophic biofloc with probiotics, heterotrophic biofloc with probiotics, and this is a flow-through clear water system. And then after growing them in the floc, they moved them from biofloc to seawater, so that's a clear water, or biofloc to biofloc, basically back into the same water after the challenge with Vibrio parahemolyticus. And then they brought in shrimp that had not been previously exposed to biofloc and then brought them in and put them into these bioflocs. And this is the survival results from that challenge. And you can see biofloc to biofloc, you get really good survival, kind of across the board here. And the biofloc to seawater, so basically it went from floc to clear water, you had pretty low survival, so they lost whatever protection they had and succumbed to the challenge. They died from that challenge with Vibrio parahemolyticus. What was really interesting, however, was the ones that didn't have any previous exposure, that were held in a recirculating system and then brought into the bioflocs, they also had pretty good survival. So it was really difficult to explain this result and to see what was going on. So they did a follow-up study and to try to examine what is the mechanism that's going on. And basically what they found out was, and they set the experiment up more or less the same way, and basically what they found is that the biofloc induces this phenotypic switching, okay, from a pathogenic form to a non-pathogenic form. All right, so remember, that's what I talked about. The Vibrios, they can be good Vibrios or bad Vibrios depending on this quorum sensing, on this communication system. And biofloc basically is a part of that, okay? And in biofloc, it offers some protection. It basically shuts the genes off for pathogenicity in Vibrio parahemolyticus. So that was a really interesting result and really shows the value of having some floc in the system at controlling the pathogenicity of Vibrio parahemolyticus. Okay, similar to biofloc, probiotics also have beneficial impacts on environmental quality and on host status with respect to growth and the immune system and having a balanced system, okay? And again, it can downregulate this virulence gene if you have the right kinds of probiotics in the system. So again, similar kind of dynamic that you'd see with biofloc. So here on the right, I've got a whole list of probiotics that can be used to control vibriosis, presumably. And there are a list of mechanisms of why they do that. They compete with Vibrio. This is inside the intestine. These bacteria produce these antimicrobial peptides. These things called bacteriocins. They're kind of natural antibiotics is one way to think about it. You know, there's always, these bacteria are always warring against each other. Those are those red lines in that interactive community graphic I showed you earlier. They produce organic acids. So actually for me, this is one of the feed additives that I think really works well is organic acids. A lot of the shrimp farmers in Latin America are using organic acids to lower pH in the gut, which is not favorable for Vibrios. Some of these produce chemicals that block quorum sensing. They're quorum sensing inhibitors, or quorum quenchers is another name. Some form biofilms, and some shrimp stimulate the immune system. So you can see there's a variety of mechanisms involved here. Okay, so if you look out there in the world of probiotics, and it's a pretty dynamic space right now, you can divide the kinds of bacteria or the kinds of products out there into those bacteria that produce lactic acid, those that don't produce lactic acid, various yeasts. There are others in addition to this one. Photosynthetic bacteria like the rotobacters, we have nitrifying bacteria and denitrifying bacteria. So there's a lot of different kinds of probiotics out there. And typically if you buy a probiotic, you're buying a mixture. You're going to have a number of these present in the product that you buy. So what I want to do now is just go through a few of these species, and probably the most common probiotic out there on the market right now are various bacillus species. You know, one of the reasons why bacillus is so commonly used as a probiotic is that it produces spores. You know, for those of you who work in shrimp hatcheries, you know about Artemia, and you buy a can of Artemia cysts, and then you hatch those cysts. It's kind of like that. You know, you basically have these spores. The spores are stable. They're resistant to desiccation and cold and so forth. And you basically germinate those spores, and then the bacteria become active. So they're very stable and easy to use. The other point to make here about bacillus is that, you know, for me, one of the main benefits of bacillus is that they are big enzyme producers. They are like enzyme factories, okay? And they produce all kinds of enzymes that are beneficial, proteases and so forth. So as the feed is moving through the gut, and remember, the retention time in a shrimp gut is very short. It's half hour, maybe an hour at the top. And so, you know, having those bacteria helping you with digestion can help improve feed conversion, which is usually important. You know, they have other roles too. They produce these antimicrobial peptides, these bacteriocins that I mentioned before. They stimulate the immune system, improve growth performance, really primarily through these enzymes. And then they can also function as bioremediators, and I'll get more into that a little bit later. One important point to make, this is something we were discussing a little bit earlier before the presentation, is that these probiotic bacteria have different capacities. So if you look at Bacillus probiotics, you know, they produce enzymes, they have antimicrobial activity, they produce these compounds, they can decompose organic matter, some do denitrification, some do flocculation. The capacities of Bacillus to do these things varies. It varies by species, and it varies even by strain. So here we've got 7 different strains of Bacillus subtilis, a very common Bacillus probiotic. And you can see that the enzyme production, the height of those bars, varies quite a bit. And in particular, I wanted to focus on cellulase. That's this green bar here. So this particular number 5, this strain number 5, is one that I would probably want to use in my shrimp pond. Why? Because the most common organic matter that you're going to find in there is algae. And you want a cellulase to break down the cellulose, that is the plant material, that is that algae, okay? So you can see the height of the green bars varies quite a bit across these different types of bacteria. So it's important to know the performance. Not all bacillus are the same. Okay, again, I mentioned this, these bacillus have really broad roles, and they can serve as probiotics. They can do this quorum quenching, interrupt this signaling. They can degrade these signal molecules, for example. And then they can act as bioremediators to aid the decomposition of organic matter in a pond. Okay, one of the other more common probiotics are these lactic acid bacteria. And for me, these are really true probiotics. These are ones that have a real beneficial impact internally. And again, there are a whole number of good candidates for lactic acid bacteria. And again, in terms of fighting Vibrios, they act very well, principally by lowering pH and producing these bacteriocins. So they're commonly used, and they're also a spore former, like the bacillus. One of the lactobacilli that I think is really interesting is this Pediococcus, and you can see it has a variable metabolism, and it's very common in these fermented foods. And this bacteriocin here, this pediocin, is very powerful against Vibrio. If you look over here, so here's the percent viability of Vibrio parahemolyticus with all these different strains of bacillus, and here's Pediococcus. And so really it reduces the viability by about 80%. So that's a very potent tool that you can use to fight against vibriosis. This is a really interesting bacteria, and I know it's used quite a bit in China, and I actually encourage wider use of this bacteria. And remember that in one of the graphics I showed that the healthy shrimp had rotobacters in their intestine. Now it's really difficult to apply this in the feed because it's a liquid form. It's not a spore-forming bacteria like the bacillus or the lactobacillus, but it's incredibly diverse. It can grow with or without oxygen. It can get energy from sunlight or inorganic or organic compounds. It can get carbon from inorganic carbon or organic carbon. It's got this beautiful color, but it actually has a very bad smell. It's a good one to grow. And it also here, let's see, where's the rotobacters? Down here. Some are actually pretty good as denitrifiers. Okay, so this is one that's usually found in anaerobic sediment. You can probably just isolate it. If you have a specific media, you can streak plates. You streak plates a few times, you can isolate colonies and grow them up. Just play with the media a little bit. Again, widely used in China. I think we need to see more application of this as a probiotic. Paracoccus is an interesting one. If you have a problem in your shrimp ponds with black gills, that usually indicates anaerobic conditions in the sediment and the presence of hydrogen sulfide. And this Paracoccus will basically oxidize the hydrogen sulfide. So what this graphic shows is the hydrogen sulfide concentration over time. And here's the control, and here's this one particular product, and you can see very quickly it reduces the hydrogen sulfide in the system. So this is a good one for that particular purpose. If you've got a hydrogen sulfide problem, apply some Paracoccus, it's going to solve the problem for you. Okay, and then I've listed this particular yeast. Again, there are other yeasts out there. I was just reading this morning about a yeast called Pichia. that seems to have some interesting properties, so I'll look more into that. These are used typically for baker, brewer, or wine making. It's basically a fermentative organism, and really the part of it that, the cytoplasm is beneficial, but really it's the cell walls that are very active, and there are these mannine oligosaccharides, which is a prebiotic, and so there are definitely benefits to using that as well. So that's another probiotic to consider. Okay, and then the last tool I just wanted to mention briefly are using fermented supplements. And I know a lot of farmers now have an interest in applying fermented rice bran, but really any material is suitable for fermentation. The thing I would say about using fermented rice bran is one of the key aspects is to use a very, very fine particle. And you should screen the rice bran so that you have particles less than 200 microns. Why do you want to do that? Because when you add it to the pond, if you have particles greater than 200 microns, they're going to just settle down to the pond bottom and create problems. But if you put less than 200 microns, they very easily stay in suspension. And so you want that material in the water column to provide this probiotic benefit. And so there's a lot of benefits here. It provides this flush of the intestine. You can use it to treat water. You can use it to stimulate zooplankton blooms. And then you can use it to aid in decomposition of organic matter. Some of the farmers I work with in Latin America are using fermented soybean meal. And basically the way they do that, just very briefly here, it's a two-step process. The first fermentation is liquid fermentation, and basically you do that for a day, okay? So that's sort of the mother culture, the liquid culture. And then you mix it on a one-to-one basis. So 1 kilo of soybean meal for every liter of this liquid, and then you put it in bags for another day, okay? And then it's ready to feed on the third day. And the farm that I've worked with on this, what they do is they use this as the first feeding every day, okay? And they start feeding around 11 o'clock, and they feed this fermented soybean meal one time. And then at 12 o'clock at noon, they turn on their automatic feeders, and then they feed up to midnight, basically every 10 minutes. But this soybean meal acts as kind of a flush. It sets the stage for good utilization of the feed that follows. This farm has been able to achieve, especially when prices were poor, they were able to replace feed up to 20%. I say all this because it's soybean meal, and this is a USSEC event, and so this is a good use of this material in addition to adding it in feed. All right, in the last part of my presentation here, I just want to go through some aspects of bioremediation. And the first thing I want to say is that I think we should be a little bit specific about words that we use, okay? For me, a probiotic is something that is related to the internal health of the animal, okay? That's something that you're adding with the feed, and it's consumed, okay? And the intention is to stimulate the immune system, provide digestive enzymes, that sort of thing. Whereas bioremediators are maybe the same bacteria, okay, but you're applying them for a different purpose. You're applying them to the water, you're applying them to the soil with the idea of improving those. And specifically you're applying them to the soil to digest or improve or decompose, aid in the decomposition of organic matter in the soil. So let's try to use the specific word for the specific purpose, even though they might be the same bacteria. Okay, so when you buy these products, they come in different forms, all right? And each form has its benefits and its costs or its pluses and minuses. Liquids are interesting. They're basically ready to use. the problem is that they're mostly water. So if you have a transportation issue, that's a cost you have to bear. Now, it's really maybe the only choice for nitrifying bacteria there, and rhodobacter, I should add to that one as well, just because they're in that liquid form, and there are no spores. Powders, again, going back to the spore-forming bacteria, you can buy bacillus and lactobacillus in powdered form and simply activate them. And that's all that's needed, is just activation with water. You can also add it to other sources of carbohydrate to aid in activation. And there are these biodegradable, these starch-based plastics that can be used, and you basically just toss it into the pond and you're ready to go. There are also these types of probiotic forms that are much bigger, and I actually like them as bioremediators because you put them in and they go right down to the pond bottom. If you have an area of the pond, let's just say near an automatic feeder, and you put these little tablets or discs down there, those bacteria can germinate and go to work right where you need them to go. Okay, so it's a very directed way. Okay, so this is an example of a program that you might want to use if you have a lot of organic matter in the water. And farmers often frame this concern to me this way. They say, I've got too much algae, I've got too much blue-green algae. What do I do? So basically there's a competition between algae and bacteria for all kinds of resources in the pond. And normally algae is going to win because it's exposed to sunlight. So you have to kind of force it over towards a more bacterial system. That's why you add a little bit of organic carbon to the pond. So you can add these materials to basically act as oxidants. You can use, there are other oxidants out there that you could use. Potassium permanganate comes to mind. And then you want to come back in with these other bacteria. So these photosynthetic bacteria are the rhodobacters I showed you, and then heterotrophic bacteria like Bacillus probiotics. And then if you have water with high turbidity, you want to add some kind of coagulant. You can use this PAC, you can use gypsum, you can use alum. So there are a number of materials that you can use as well. But again, the idea is to try to shift the competition a little bit more over towards the bacteria. And you notice I didn't have any organic carbon addition in this scenario. So you're basically just trying to inoculate with bacteria. Now, in terms of soil bioremediation, again, I work with farmers that have maybe a little bit different context than here in Thailand, where things are a little bit more intensive. But for farms that have earthen ponds or earthen bottoms, soil bottoms, one of the tools that is really useful is this thing called a triangle. And you basically throw that triangle out and you pull it back in. And you look at the mud, you look at the soil, and if it looks like this, if it has a nice brown color, then you have good condition. This is what you would have. And you might add some probiotics once a week or so. But it's really not necessary. If you get down here where you have very low oxygen and this black soil has a very foul odor like rotten eggs and shrimp are dying, then you probably want to add bacillus as a bioremediator and lactobacillus as a true probiotic, and then add this paracoccus to reduce the hydrogen sulfide. So depending on the conditions, you would adjust your program as well. A lot of farmers ask me this question too about what do I do between cycles? And farmers never want to have their ponds being down out of production for very long. Now this applies to earthen ponds. If you have a plastic liner context, this obviously doesn't apply. You can turn things over very quickly. But normally you want to try to dry it if possible, and then add some lime. And particularly in the context now with EHP being out there, you can add enough hydrated lime or burnt lime to raise the pH above 12 to activate the spores before you stock. Okay. But if you're really focusing on organic matter, then you can just simply apply some bacillus. If you want to do a rapid turnover, you can basically do the same thing, this bioremediation, and then add some nitrogen, because often that will be a limiting factor in decomposition of organic matter that accumulates in the system. A lot of farmers now are using automatic feeders, and you can see what happens. You know, you get this accumulation of organic matter right below the feeder during the production cycle. So I encourage farmers to apply bioremediators during the production cycle. You don't have to wait until it's over. Add it during a couple times a week or a few times a week, depending on your circumstances, to make sure conditions are okay. I mentioned this earlier. If you have problems with black gill, usually indicating a hydrogen sulfide condition, you can apply this Paracoccus bacteria as a bioremediator as well. Okay, and these are really a basic set of conditions that you need to consider with respect to a bioremediation program. You want to make sure you select the right organism, and most of them, as I said, are mixtures. They're going to have different species and different strains that are going to, each is going to do something for you. Normally, you should have products that are going to give you this effective microorganism concentration of at least 10 to the 8th. Some even guarantee up to 10 to the 10th, and most products will give you that. I encourage farmers to use the activation protocols that are suggested by the manufacturer. A lot of farmers have their own kind of recipes for using probiotics. You know, what I've seen is that if you don't add it, if you don't run through the proper protocol, after about a day, you see a big divergence in what you actually end up applying to the pond. So activate and apply. Just do it quickly, followed by the instructions. And then the conditions that you store that product should be just like this room. Should be cool, dry, and try to use the product quickly, okay? But if you're going to store it, keep it in good conditions. Now, there are some limitations of using these bioremediators. Most ponds already have very high concentrations of bacteria. And often it is not the absence of a particular strain or a particular quantity of a bacterial population, but it's something more fundamental. Temperature, and specifically dissolved oxygen. If you don't have enough dissolved oxygen in the pond, you can add all the bacteria you want, but without that oxygen to help those bacteria work, you're not going to achieve anything. Sometimes the products don't contain the type of bacteria species at the indicated loads. And this reminds me to say something here that I learned recently about informal probiotics that were being marketed in India. And those informal products were tested and found to contain genes for antimicrobial resistance. So farmers, by replicating these bacteria, were contributing to the problem of antimicrobial resistance. I was happy to learn this morning that Thailand requires, as a condition of registration of probiotic products, that they be tested for the presence of these kinds of genes. So that's a good thing. So buy products from reputable suppliers, not from somebody who's isolated it from your shrimp pond and he's replicating it in his laboratory, his little homemade laboratory. Get registered products. Don't contribute to the problem. This is an important point too. Every pond is different. You know, I've made a lot of general statements here today, but really the underlying point should be go and look at what conditions are on your farm and make adjustments to your applications based on what you see. Can you advance the slide, please? Okay. So just 3 final slides here to just kind of wrap this up. These are the messages that I want you to leave here with. I know I presented a lot of information to digest. You might need to take your own probiotics to digest this information. But let me try to summarize briefly here. Going back to the discussion of these, of the microbial community itself, these random processes are really important. And so what that means is that, you know, these communities are really dynamic. They're changing all the time. And it makes it really difficult to try to manipulate that to your benefit. You know, many farmers think, okay, if I add Bacillus probiotics to the feed, I'm going to find it in the gut. Well, sure, you can actually find some in the gut, but not at the numbers that you might think based on how much you add it to the feed. It doesn't really colonize. What I think happens to the Bacillus is that it comes through the intestine with the feed. It works to produce these enzymes to aid in digestion. That's a good thing. Does some immune stimulation. That's a good thing. And then it leaves with the feces. And then it becomes a bioremediator. And it aids in the decomposition of that organic matter, that solids. So I think it actually serves both a probiotic role and then subsequently a bioremediator role when it leaves the animal. But the colonization isn't really a big thing. So the microbial community shifts in response to disease. This is this idea of dysbiosis or imbalance in the system. It's almost diagnostic. I mean, there's obviously easier ways to diagnose disease, but you can actually tell that the animal is sick based on the microbial community. And we're really not at the point where, you know, this type of analysis that requires next generation sequencing is going to be routine on farms. This is really an academic or level at the university. Okay, I mentioned that Vibrios are keystone species. They are these opportunistic pathogens, but they're important. They hold the community together. So the effort should not be to try to get rid of Vibrio. It should be to live with them and to keep their genes for pathogenicity turned off in whatever way that can be achieved. And mainly, the key risk factor is organic matter accumulation. So keeping pond bottoms extremely clean is really important. Okay, and going to our tools now, we have this microbially mature water. You know, I think this is probably, of the tools that I mentioned, this may be the one that applies most, I think, or easiest, can be easiest to implement on farms. So have this microbially mature water. Biofloc systems and probiotics really kind of help with the same idea where you're trying to suppress this pathogenicity, keeping those genes for pathogenicity turned off. And I also mentioned about fermented supplements, and those also can be beneficial as a tool. All right, and then finally, again, talking about the different words that we're using, probiotics are for improving intestinal health, and bioremediators are to improve water and soil quality. So, you know, we can add these activated bacterial preparations along with fertilizer, nutrients, and liming materials. And I'm sure this is true in Thailand now too, with a lot more use of automatic feeders. And with these automatic feeders, you've got a lot of organic matter accumulating beneath them, so focus your efforts there for bioremediation. And I mentioned just briefly about that these bioremediators, sometimes they don't work very well. And you know, the farming systems that I've worked with in Latin America, compared to what you have here, are much more extensive. And really, they're semi-intensive farms. They're stocking mostly between 10 and 15 per square meter, maybe up to 20 per square meter. So a fairly low level of intensity. And farmers will tell me, well, look at my soil. It's beautiful. It's brown. It's not black at all. I said, that's great. How much were you able to increase production? I don't know. And so it didn't really have a benefit. But when you have an intensive system with aeration and automatic feeders and all kinds of other technology, a whole package, probiotics or bioremediators are essential. So as systems become more and more intensive, the use of bioremediators can be more beneficial to the farming system. Okay, and with that, I thank you very kindly for your attention.